Welcome to the lecture series dealing specifically with metabolic reactions within a cell or an organism. These lectures also will include discussion of energy, catalysis, and biosynthesis. But the first thing I want to do is define the word metabolism. You may have some preconceived notion of what metabolism means. Oftentimes we think about metabolism when we talk about weight loss or an individual's ability to lose weight. However, the true, accurate biological definition of metabolism is it is an organism's complete set of chemical reactions. That is, all of the chemical reactions that occur within a cell or an organism is collectively referred to as metabolism. In the figure that you see on the left-hand side, this is an example of some metabolic reactions and really meant to just emphasize the complexity of metabolism. In this figure, again on the left, you see a few hundred reactions out of the thousands of reactions that take place in a cell. The dots in the diagram or figure represent molecules, and the lines between the dots represent chemical reactions transforming those molecules. Now, because metabolism is such an all-encompassing term, again, it refers to the complete set of chemical reactions that are occurring within a cell or an organism. We like to break those down into two broad categories. There are metabolic reactions that are referred to as catabolism, and catabolism or catabolic reactions is defined as the breakdown of large molecules into smaller compounds. With catabolic reactions, you typically see a release of energy. A great example of catabolic reactions or catabolism is the process of cellular respiration. We'll talk about cellular respiration in a few lectures down the road in a lot more detail. But in general, when we talk about cellular respiration, this is a process where glucose and other food or fuel sources are converted into carbon dioxide and water. Energy stored in the glucose molecules is released to then do work inside of the cell. So this is a great example of breakdown of larger molecules into smaller compounds, and associated with that is the release of energy. Energy that is stored in those larger molecules. So in the diagram or the figure on the right-hand side of the slide, particularly focus on the left-hand portion of that diagram, you can see up at the top the larger food or fuel molecules that are then broken down by catabolic pathways into smaller components that can be utilized in the cell. The important thing here to note is that, again, useful forms of energy are typically released in these types of reactions. In addition, there is a great amount of heat that is lost when these reactions take place, but we're really not going to focus on that for our purposes in this cell biology course. So the other term when we talk about metabolism is anabolism or anabolic reactions. These types of reactions are almost the flip or the opposite of what we just discussed with catabolic pathways or catabolic reactions. Anabolism is the conversion of small, simple molecules into larger, complex molecules. These types of reactions typically consume energy or require energy for the buildup of these large molecules. So again, if you focus on the figure on the right, this time looking more at the right-hand side of the figure, you can see starting at the bottom these smaller building blocks that are utilized in anabolic pathways or anabolism to then build larger molecules that the cell requires. But in the very center of that figure, you can see that energy is required as an input to build or synthesize these larger molecules. So what you see in this figure is a great example of the unique give and take relationship that occurs between these two broad categories of metabolism. Catabolism is an energy releasing type of reaction. The energy released there is then harnessed and utilized for those anabolic reactions that require the input of energy. Now, before we continue talking about metabolism and metabolic reactions, we need to take a few steps back and review some basic concepts that come from physics. One of those concepts that I've already mentioned in the terminology I used in the previous slide is the word energy. Energy is defined as the capacity to perform work. So it's the ability to do work, which is really perform any type of activity. Move things around, rearrange things, mechanical transport, all of these things are considered work. And so energy is defined as the capacity to be able to perform that work. Just like we talked about in the previous slide, the term metabolism is a very large, all-encompassing term. Energy is also a rather large, all-encompassing term. We can break energy down into two basic forms. There's what's known as kinetic energy, which is the energy of motion. Anything that moves possesses kinetic energy. Additionally, there is potential energy, and potential energy is a stored form of energy. One of the best examples, taking it again away from cell biology, is from the figure that you see here on this slide. What you see is a dam, and on one side of the dam you see the water that is being held back in the reservoir, where the dam is basically blocking the flow of the water. You can also see the floodgates or the dam gates opened where water is being released. So in this particular figure, the water in the dam, the one that's in the reservoir behind the dam, has a great deal of potential energy. It has the potential to do something. It is a stored form of energy. Likewise, the water that has been released through the dam floodgates has kinetic energy associated with it. That water is in motion, it is moving, it is dumping out from the reservoir down into the valley below. And again, this has kinetic energy. This energy can be utilized to perform some type of work. And we'll come back to that concept in just a little bit. But tying it back into cell biology, I want to talk about something known as chemical energy. Chemical energy is a form of potential energy that is stored in molecules due to the arrangement of the atoms in those molecules. And more importantly, this energy can be converted into kinetic energy. So molecules do in fact have typically a great deal of potential energy associated with them, due again specifically to the arrangement of the atoms in those molecules, and energy can be released and utilized in the cell in a specific way. So now we're going to continue with some basic physics principles and discuss a few additional components because they are essential to our understanding of what's happening in metabolic reactions. Another set of terms that I want to introduce, or the first term I want to introduce, is something that's referred to as free energy. So this is a portion of a system's total energy that is available to do mechanical work. So this is specifically what we're interested in from the cellular point of view, is the ability to do some type of work. So the portion of the energy that can participate or contribute to that is what we're really interested in, and that's referred to as the free energy. That is also designated as the capital letter G. G is equal to the total amount of free energy in a system. Now, what we are really interested in from the basic physics point of view, but also from the cell biology point of view, is the change in free energy. And that change in free energy is designated as delta G. So that little triangle before the capital letter G is the Greek letter delta, and this really just means the change in free energy. So again, what we're really interested in is the change that occurs with reactions to help us understand what's happening with the release of energy that can then be utilized in the cell for other purposes. So the delta G, or the change in the free energy, is defined as the G of the final state minus the G of the starting state. And we'll go over that in a little more detail with this figure and in the next slide in just a minute. So let's look at this figure in a little more detail and hopefully get an understanding of what's happening in the changes that are occurring from the initial state to the final state. So in each one of the components of this figure, the top portion of the diagram represents the initial state and the bottom portion of the diagram represents the final state. So let's look on the far left-hand side of this figure, which is labeled gravitational motion. What you have is a child up at the top of a slide. This instance or this example has a great deal of potential energy associated with it. It is extremely unstable, and gravity will draw the child down the slide to the bottom. What happens when the child reaches the bottom of the slide? This is a much more stable state. There's really no fear of the child falling off the slide at this particular point. But what has happened is there's been a change in the free energy, in that the child at the top of the slide has a great deal of free energy associated with it, and this is extremely unstable. And the child at the bottom of the slide has very low potential energy associated with it, and this is a much more favorable situation. Now, the next two examples on this particular figure are a little more biologically relevant. What you're looking at in the center is an example of diffusion, and then on the far right is a sample or an example chemical reaction. So let's focus on the process of diffusion, which we talked about previously. We know that diffusion is a mechanism that occurs when you have a concentration gradient with a molecule of higher concentration, and in this case separated by a membrane that is permeable to that molecule, and a lower concentration of that molecule on the other side. That particular figure at the top of this diagram shows a situation where there is high potential energy. This is an unstable situation, and what will happen eventually is those molecules will diffuse across the membrane to reach an equilibrium state. This is much more stable, much more favorable, and has a lower potential energy associated with it. Similarly, the example on the right-hand side is a chemical reaction. In this case, you have a more complex molecule that has a high potential energy associated with it. This is unstable, and at the end of the reaction, you have smaller molecules that have been formed. These are much more stable and have lower potential energy associated with them. Equilibrium is defined as a state of maximum stability. Most reactions are reversible and proceed until the forward and backward reactions occur at the same rate. As a reaction proceeds towards the equilibrium, the free energy decreases. Chemical equilibrium is similar in concept in that there's no further change in the concentration of reactants or products. Chemical equilibrium would be defined where a delta G is equal to 0, meaning there is no net change in the system. However, when we're talking about chemical reactions, we are oftentimes interested in chemical reactions with changes in free energy, and those fall into two different categories. The category on the left is referred to as an energetically favorable reaction, and the one on the right is referred to as an energetically unfavorable reaction. The energetically favorable reactions are also referred to as exergonic, and these reactions, energy is released. The delta G is less than 0, and again, these are energetically favorable with energy being released. So let's look at the diagram in a little more detail. This is referred to as a free energy diagram, and it will come up in several of the slides coming down the road here. So we want to make sure that we really understand what we're looking at with a free energy diagram. With a free energy diagram, you have the progress of the reaction along the x-axis, and you have the amount of potential energy or free energy of the molecules involved in the reaction along the y-axis. So in this energetically favorable reaction, you can see that there is a certain amount of potential energy associated with the reactants. At this point, it doesn't really matter what that value is. You just see there is a certain amount of free energy associated with the reactants. What you can hopefully clearly see in this figure is that the free energy amount or level associated with the products is lower than that of the reactants. In this case, that change that is occurring from the free energy of the reactants to the free energy of the products is the amount of energy output that is being produced from this reaction. This is also sometimes referred to as a downhill reaction. Again, you're going from a level of free energy associated with the reactants and you're moving down towards the products. This is a delta G less than 0. And again, if we go back to our definition of delta G, the delta G is equal to the G of the final state minus the G of the starting state. So it would be the G of the products minus the G of the reactants. If you now focus on the figure on the right-hand side, this is again an energetically unfavorable reaction. It's also referred to as an endergonic reaction. In this type of reaction, an input of energy is required, and the delta G is greater than 0. So again, this is a free energy diagram with the progress of the reaction along the x-axis and the amount of potential energy or free energy of the molecules on the y. So here what you're looking at is a certain amount of potential energy or free energy associated with the reactants, and the amount of free energy or potential energy associated with the products is at a much higher level. This is an uphill type of reaction, and there is a requirement for an input of energy in order for these types of reactions to proceed. The next set of figures on this particular slide is actually one of my favorite ones to try and draw comparisons to what's happening in chemical reactions within the cell. So let's break this figure down and hopefully be able to draw those conclusions or those comparisons between these reactions in this figure and what's happening in a cell with biochemical reactions. So let's focus on panel A first. What you're looking at is what's referred to as a closed hydroelectric system. You have a system where you have a turbine in the center, and if water rushes past the turbine, it'll turn the turbine and it'll generate electricity. Now, what you might ask yourself is, what does that have to do with cell biology, generating electricity? Well, it really doesn't. But it really drives the point home that, again, we'll hopefully make those comparisons to within the cell. So just bear with me as we go through this example. So let's look again at panel A. Panel A, the left-hand side of panel A, you have a very simple system. You have a tank with water in it. You have another tank with water in it at a lower level than the first tank. And in between, you have a turbine that can turn, and if that turbine turns, it's going to generate electricity to turn the light bulb on. In this very simple system, as long as that water in the first tank is full, and there's an opening or a connection between the first tank and the second tank, the water will rush from the first tank, turn the turbine, generate electricity, turn the light bulb on. This is a delta G that is less than 0, but this is also a closed system. What's going to happen when all of the water rushes out from tank 1 to tank 2 and eventually levels out? You reach a level of equilibrium of the water. Now, the turbine can no longer turn. There is no more water rushing through with its kinetic energy to turn the turbine to generate electricity. We've reached equilibrium. This is a delta G equal to 0, and we no longer have energy being released. There's no more turning the turbine to generate electricity. If your goal was to continue to have that light bulb on, this is not a very well-designed system, because once you reach equilibrium, there's no more turning the turbine, no more electricity generated. So how can we make this system better? In panel B, we look at a way that this system can be made better, and by better, still allowing for electricity to be generated. Same tank system, first tank, turbine in between the second tank at a lower level than the first. But the modification to this example is that we now have a faucet that's always turned on. So that means we have a constant supply of water coming into tank 1. And we also have a valve that we've opened in tank 2 to let water be released. So as long as there is water coming from the faucet to fill tank 1, there will be enough water to pass the turbine, turn the turbine, generate electricity. This is an open system. There's a constant input of water into tank 1 and a constant release of water from tank 2. So we can argue that this is a better designed system if our goal is to continue to always turn the turbine and generate electricity. This is a better system than the closed system we looked at in panel A. All right, so now let's look at what is panel C down at the bottom. This system can be made even better. And by better, again, if our goal is to generate electricity, what if we made this a multi-step open system? We now have several tanks. Each one of the tank is at a slightly lower level than the one before it. There is a turbine in between the two tanks, or in every example. And this is an open system. Water continually coming into tank 1, water continually being released from tank 4. By making this a multi-step open system, we've now turned 3 turbines, generated enough electricity to turn on 3 light bulbs. And so we can argue again that this is a much better design if our goal is to generate electricity to turn on light bulbs. All right, so now let's move ourselves back into cell biology and think about biochemical reactions in a closed system, in an open system, or a multi-step open system. If the goal is to release as much energy from a molecule as possible, it would make sense to do it in a multi-step fashion so that as much energy that could be extracted is extracted, and also to have it be an open system where there's a continual source of fuel and there's a way to release the excess. This is exactly what's happening in cells in chemical reactions. There are multi-step open systems that allow for the maximum amount of extraction of energy possible. So now that we have a good foundational understanding of what's happening in changes in free energy in typical metabolic reactions, what I want to do is talk a little bit about enzymes. So metabolism, as I defined at the beginning of the lecture, is the total amount of reactions that occur within a cell or an organism. Now, most metabolic reactions require the assistance of enzymes, which are catalytic molecules that change the rate of a reaction without being consumed. If you recall, when we talked about macromolecules, specifically proteins, we talked about how proteins were extremely diverse, and one way we can deal with that diversity is to categorize those molecules. One category were enzymes, and I said we would come back to those a little bit later in the semester. So enzymes, again, are catalytic molecules that change the rate of a reaction without being consumed. If you look at the figure on this slide, what you're looking at is the catabolism of sucrose. What you see is a hydrolysis reaction where sucrose, which is a disaccharide, is being broken down with the assistance of water, because again it is a hydrolysis reaction, to form glucose and fructose. There's a breakage of the bond between those two monomers in sucrose, and there are new bonds that are forming with the water that is being utilized in these reactions. Now, this particular reaction is a reaction that does in fact normally occur, but the reaction time for this to take place is in the order of years. However, when you add the enzyme that helps catalyze this reaction, the reaction takes a mere seconds compared to years without the help or the assistance of the enzyme. So what I'd like to do is add a few additional terms and set the stage for discussion of dealing with enzymes. And that is, the first thing I want to mention is that enzymes, again, speed up chemical reactions, but do not get consumed in those chemical reactions. When you are looking at basic chemical reactions, you should by now be familiar with the fact that when you look at an equation or a chemical reaction, the components on the left-hand side of the arrow are known as the reactants, and the components on the right-hand side of the arrow are known as the products. Additionally, there's the term substrate. The substrate is the reactant an enzyme acts upon. Additionally, the enzyme name is typically written above the arrow of the reaction or the yield indicator of the reaction. And then in addition to that, we can talk a little bit about the enzyme and substrate interaction. For a long time, it was thought that enzymes and their substrates interacted like a lock and key mechanism. Now, that lock and key mechanism is not entirely incorrect because there is a specificity of an enzyme for its substrate. So any enzyme cannot just act on any substrate. There is specificity associated with that. However, we now know that there is more of an induced fit model that occurs. So it's not a fixed rigid structure like a true lock and key, even though the specificity is important. We know that the enzyme and the substrate interact with one another. Specifically, in the enzyme, you have what's known as the active site. And the active site is the part or the region of the enzyme that the substrate binds to. And we know that typically enzymes will interact with their substrate and sort of close down or hug down more tightly in association with the substrate or substrates. So hopefully you can see that in this particular figure with the tighter binding of the enzyme around the substrate once that interaction has taken place. Now let's look a little more closely at how an enzyme works overall. If you look at this figure, you have several steps that are associated with a pathway that an enzyme is involved in. Up at the very top, you have step 1, where you have the substrate entering the active site. And in this particular case, we are looking at the enzyme known as sucrase, and its substrate is sucrose, like the reaction that we looked at just a few slides ago, where sucrose is going to be broken down into individual monosaccharides, glucose and fructose. So again, step 1, you have the substrate binding to the active site of the enzyme. And again, you have the induced fit where the enzyme sort of closes down more tightly around the substrate. In this cycle, step 2, the substrate is converted into the products. So we said or mentioned previously that this is a hydrolysis reaction. Water is also going to be utilized in allowing for this reaction to proceed, but the bond is broken between those individual monosaccharides to produce glucose and fructose. Step 3 then, the products are released. And then step 4, again working in this clockwise fashion, the active site is now ready for another molecule to enter. So this again demonstrates how enzymes are not consumed in the reaction. They're just playing a role to help speed up those reactions overall. The average enzyme will catalyze about 1,000 reactions per second. So these are pretty efficient, fast-working molecules to allow for 1,000 reactions per second to take place. And again, that's the average for enzymatic activity. So the next question that may have entered your mind is, how is it that these enzymes are actually speeding up the reactions that are occurring? Well, enzymes can do a number of different things. Typically what they do is help put the substrate or substrates into the proper orientation for a reaction to occur. So one way is that they may help stress, stretch, or bend the bonds to help with the breakage of the bonds if you're talking about a catalysis or a breakdown type of reaction. Another way is that the enzyme may provide the right microenvironment for the reaction to take place. If you have a reaction that needs a specific type of environment, let's say an acidic environment, the active site may be lined with acidic amino acids to help provide a pocket of acidity for the reaction to proceed. So this is, again, an example of providing the right microenvironment. Very rarely there are instances where the enzyme may have a direct participation role where covalent bonds can form between substrates during the reaction, but this is again very rare, and the enzyme again is not consumed in the reaction. So again, typically placing the molecules in the proper orientation for a reaction to occur is the number one way that enzymes help speed up biochemical reactions.